Exploring Life. Chapter 14, Pattern of Inheritance. Part 2. While Mendel's model is a great starting point, real-world genetics is much more complex, and most traits don't follow simple dominant and recessive patterns. We'll explore some of the most common types of complex genetic interactions next. Multiple alleles means that a single gene has more than two possible versions, or alleles, within a population. A classic example is rabbit coat color. The coat color gene has 4 different alleles, which results in a wide variety of coat colors in rabbits. Even though an individual rabbit can only inherit 2 alleles, the presence of multiple options in the population increases genetic diversity. This image shows an example of multiple alleles in humans. For the gene that determines surface sugars on red blood cells, also known as the ABO blood group gene, there are 3 different alleles, IA, IB, and I. Incomplete dominance occurs when neither allele is completely dominant over the other. As a result, heterozygous individuals show a blended, intermediate phenotype. An example is seen in African violet flowers, where crossing red and white flowers produces offspring with pink flowers. Codominance occurs when both alleles in a heterozygote are fully and equally expressed. Neither one is masked or hidden. Some African violet flowers may appear to show codominance, with patches of different colors. However, this is not a classical example. Their coloration results from more complex genetic and environmental interactions. The true codominance well-known example is the ABO blood group system. This image illustrates codominance using the human ABO blood group system. The gene responsible has 3 alleles, IA, IB, and I, which determine the type of surface sugars present on red blood cells. When both IA and IB are inherited, the result is type AB blood, where both A and B antigens are fully expressed. This is a classic example of codominance. Altogether, these alleles produce 4 possible blood types, A, B, AB, and O. Polygenic inheritance occurs when a single trait is controlled by multiple genes, each contributing a small effect. A well-known example is human skin color, which is influenced by at least 3 to 6 genes, possibly more, and even the simplest models translate to 64 possible skin tone combinations. Unlike simple traits, polygenic traits show continuous variation, rather than distinct categories. Pleiotropy occurs when a single gene influences multiple traits in an organism. A well-known example is sickle cell disease. One gene mutation affects the structure of hemoglobin, which in turn alters red blood cell shape. This single change leads to a wide range of effects, including anemia, organ damage, and even resistance to malaria, all tied to just one gene. Linked genes are genes that are located close together on the same chromosome. Because of their physical proximity, they tend to be inherited together, as a unit, rather than assorting independently like genes on separate chromosomes. A common example is red hair and freckles, which often appear together because the genes controlling them are linked. Recombination frequency measures how often genes are separated due to crossing over during meiosis. It helps us understand how closely 2 genes are linked on the same chromosome. If the recombination frequency is much lower than 50%, it means the genes are close together and usually inherited as a pair. If the frequency is approaching 50%, It suggests the genes are farther apart, or even on different chromosomes, where they undergo random assortment. This makes recombination frequency a valuable tool for studying gene linkage and constructing genetic maps without sequencing the entire genome or identifying every gene responsible for each trait. Mutation introduces new alleles. Sexual reproduction shuffles them into new combinations. Natural selection, genetic drift, and other forces act on these differences. Over time, inherited traits that enhance survival or reproduction become more common, driving evolution. So while Punnett squares help us understand how traits pass from parent to offspring, they also help us understand how species change, adapt, and diversify over generations. Genetics isn't just the study of heredity, it's the foundation of life's unfolding story. Now it's time to check your understanding. Use the upcoming questions or activities to test your knowledge and identify any areas you'd like to review. Remember, self-evaluation isn't about getting everything perfect. It's about deepening your understanding as you explore the science of life.